Hi, my name is DeJuana Ford. I grew up in Noonan, Georgia and went to college at the University of Georgia in Athens, where I graduated with a degree in genetics. I've been passionate about science for as long as I can remember, but I became interested in the field of human genetics in particular after my 9th grade biology teacher gave a lecture on how scientists use genetic markers to track human migration patterns. For over 10 years, my mom has battled systemic scleroderma, which is an autoimmune disorder. I've been actively involved in an organization that provides support to scleroderma patients. Watching my mother and other patients struggle to overcome the daily obstacles associated with incurable chronic illness inspired me to use my passion for science to explore questions that might improve people's lives. Working in Dr. Melissa Davis's lab as an undergraduate further cultivated my passion for the process of research and led me to seek a career in academic research. I'm most interested in the field of human genetics, especially as it relates to the study of autoimmune disease, immunology, and cancer immunology. Ultimately, I aim to have a career that incorporates academic research, teaching, and community outreach. As a researcher, my goal is to gain a better understanding of the genetic and environmental factors which drive the development of autoimmune diseases. I joined the PrEP program because I was seeking opportunities to gain additional laboratory and research experience, and also to further enhance my communication skills. These experiences will prepare me to maximize my graduate education and ultimately to be a highly effective independent researcher. PrEP at UGA's focus on infectious diseases has been especially beneficial for me as I have a variety of opportunities here to explore my interest in immunology. I look forward to gaining excellent mentorship, learning new laboratory techniques, and building upon my scientific knowledge over the course of this year as a PrEP at UGA scholar. My faculty mentor is Dr. Melissa Davis in the Department of Genetics. For the past decade, Dr. Davis has worked to gain a better understanding of the molecular and environmental factors driving tumor aggressiveness and breast cancer survival disparities. My project is focused on understanding the role that insulin-like growth factor pathway genes play in triple-negative breast cancer, a breast cancer subtype characterized by aggressive tumors that are unresponsive to hormone-based targeted therapies. Insulin-like growth factors, or IGFs, are structurally similar to insulin, but are distinct in that they associate with a set of binding proteins that regulate their activity. Recent studies have linked breast tumor aggressiveness with increased levels of IGF-2 protein expression. Prior to becoming a PrEP scholar, I, along with other members of the Davis Lab, observed that IGF-BP6, IGF-2's preferred binding partner, demonstrates distinct patterns of expression and subcellular localization in triple-negative breast cells. Specifically, we noticed that in triple-negative cells, IGF-BP6 tends to form aggregates at the edge of the nucleus and co-localize with IGF-2. This is significant because exclusion of these aggregates from the nucleus could limit IGF-BP6's IGF-2-independent pro-apoptotic activity within the nucleus. Also, IGF-2's association with IGF-BP6 might be prolonging the half-life of IGF-2, thus promoting cancer cell growth. To further investigate this pattern, I used RNA interference to reduce IGF-BP6 mRNA expression in cells derived from triple-negative tumors. To accomplish this, I extracted RNA from triple-negative breast cells and used cDNA synthesis, PCR, and finally a transcription reaction to synthesize a double-stranded RNA fragment specific to IGF-BP6. After transfecting triple-negative cells with a double-stranded RNA, I observed IGF-2 expression and subcellular localization in these cells. I hypothesized that IGF-2 would be more evenly distributed throughout the cytoplasm after the IGF-BP6 knockdown, and my preliminary data seemed to support this hypothesis. This is significant because it suggests that distinct IGF-BP6 expression patterns have the potential to impact IGF-2's cancer cell growth promoting activities. My next set of experiments will use qPCR to assess whether the IGF-BP6 knockdown in these cells impacted the expression of two anti-apoptotic downstream targets of IGF-2. Taken together, these data will give us a better understanding of the molecular mechanisms by which the insulin-like growth factor pathway might impact triple-negative breast cancer. My name is DeJuana Ford, and I am a Prep at UGA scholar.